Today's webinar is the first of the series where we will be highlighting work of Dr. Gary Miller at Emory University and a project that was funded, one of the NIEHS Core Centers that was funded last year, the HERCULES Center at Emory University. Gary received his PhD in pharmacology and toxicology from the University of Georgia, and then did postdocs both at Emory University and at Duke University, followed by faculty stints at University of Texas and back to Emory. Since 2009, Gary has served as a full professor and the Associate Dean for Research at Emory. His research interests have lied primarily in looking at the environmental contributions to Parkinson's disease, but as that has emerged, or as that has evolved, he has begun to focus largely on issues of the exposome and trying to focus on environmental contributions on a much grander scale to disease. Gary literally has written the book on the exposome. He published through Associated Press a primer on the exposome just at the end of last year, beginning of this year. And I'm sure that he will talk a little bit about that project as well as the HERCULES Center. So Gary, take it away.
Thank you, David. So let me see here as I advance. Okay. So a little bit more about me. The introduction was appreciated, but I want to point out that I am not an exposure scientist. I'm not an epidemiologist or a computational biologist or a bioinformatician. I am a mechanistic toxicologist who, as Dave pointed out, looks at environmental contributors to Parkinson's disease. But I very much believe that the environment has a more malleable effect on health and well-being than the genome. And more importantly is that the environmental side of this equation needs some better tools and some better arguments. So I want to start with some definitions, as this is the first webinar. I think we need to make sure we're all kind of on the same page here. In thinking about what we consider as health. I mean, you can look in a dictionary and we all kind of know this general overall condition of an organism at a given time. But I think that the World Health definition is probably one that's more familiar to us all, defined as a state of complete physical, mental, and social well-being, and not merely the absence of disease. So often I think in medicine we think about a very disease-centric focus, but again we're thinking about going beyond just having that absence of disease. As a scientist, I learned early on about this concept of homeostasis that was first discussed almost 100 years ago by Cannon. And the idea is that you have this steady state condition that any tendency towards change is met with a factor to resist that change. And that system consists of several cooperative mechanisms that are acting simultaneously or successively. And importantly, it doesn't occur by chance, but it is organized self-government. So keeping your biological systems in place is just not a happenstance. There's actually a coordinated system for it. Many years ago, I came across this concept of allostasis, which was presented mainly by McEwen. And briefly, the definition is maintaining stability through change, just kind of a derivation of homeostasis. And this concept of the allostatic load was presented primarily for response to chronic stress and was measuring neural and neuroendocrine responses. But what I really liked about this concept was that this allostatic load represented the cumulative cost of the correcting process. So in thinking about the sort of wear and tear on a system that comes from external insults, this is something that this concept of the allostatic load was trying to capture. But again, it was very much focused on more of a hormonal pathway. But I believe that a portion of what we'll be measuring when we look at the exposome will be some derivative of this concept of the allostatic load. We're all very familiar with the genome. You know, we have a lot of genes that have been studied for many years now. And it's been the subject of very seminal work going back to 1953 in Watson and Crick. Many of you may have heard how some of these letters from Dr. Crick to his son sold for millions of dollars along with his Nobel Prize. But again, this stands out as one of the greatest discoveries in science. And it was followed thereafter by this huge initiative of the Human Genome Project. The idea to sequence the entire human genome. We identified over 30,000 genes. But from this, we've seen that only about 10 to 30% of human disease can be explained by genes alone. And shortly after the publication of the first draft, Craig Venter, who was one of the drivers of this project, just said we don't have enough genes for the biological determinism to cover these things. It has to be more than our genes. We don't have enough complexity to do that. So while he may have been surprised by that, as someone who's been interested in the environment, that didn't really seem to be a surprise. And in many discussions, and I've heard this from Dr. Collins, who is a key driver of the Human Genome Project, is this phrase, genetics loads the gun and environment pulls the trigger. The first reference to this was by Elliot Jocelyn at Harvard. The Diabetes Center there now still bears his name, with a major focus on the manipulation of the environment as therapy. So the idea of controlling our environment for medicine has been around for over a century. But I've never been fond of this phrase that environment pulls the trigger, because it puts the environment in a very sort of passive role. Once this happens, there's nothing else the environment can do. It can just follow through on what the genes have set up. And I'll discuss this a bit more. But we know that there's a continuum between the role of genes in the environment and health. We go from the sort of classical genetic disorder like phenylketonuria, where there's a mutation in an enzyme responsible for metabolizing a particular amino acid, all the way to injuries and accidents that have minimal genetic factors. But the majority of diseases that we're concerned about, cancer, neurodegeneration, heart disease, diabetes, type 2, are definitely some sort of combination of the genetic risk factors and the environment. One thing I'd like to point out about even a condition like PKU or phenylketonuria is you have this, what would be considered a pure genetic disease. And 50 or 60 years ago, it was one of the leading causes of mental retardation and institutionalized of children. And once it was identified, what the medical field found was that if you regulated the amount of phenylalanine in the diet, you could completely reverse the damage. So an environmental manipulation was able to trump this purely genetic disease. And as we go through these other disorders, we know that how we behave and our diet and other exposures can influence that. And this is a key part of it. We have all these diseases that are kind of clumped in this gene environment continuum, and we need to really look at how we can address the environment. So many of you may be familiar with the Global Burden of Disease study that was published in Lancet just over a year ago, where they looked at diseases across the entire world. And in this figure here, looking at the risk factors for these major conditions, what's very striking about this is that when you look at risk factors for disease, you'll notice that most of these things, if you take a very broad view of the environment, are these sort of environmental factors. It's more than just the sort of classical air pollution type things, but when you look at all these things, this concept of the non-genetic factors are major drivers of worldwide disease. So the question becomes, how do we capture the environment? We know the human genome has been covered very nicely by the Human Genome Project, the HapMap, ENCODE projects. We're adding more knowledge about how our genome can be manipulated. But again, these global burden of disease risk factors are all non-genetic. And so we intuitively know that health and disease are impacted by our environment. So this brings us to this concept of the exposome. Chris Wilde coined this term 9 years ago, and he defined the exposome as all exposures from conception onwards, including those from lifestyle, diet, and the environment. And in just pointing out, this is coming from Dr. Wilde is a molecular epidemiologist. He was looking at ways of addressing environmental exposure for his studies, and this was a very insightful and visionary concept to put forward. So when I was involved in teaching my class, I was looking up some definitions on the exposome and was kind of disappointed when I looked in the American Heritage Dictionary and saw that no word definition was found. Merriam-Webster says the word you entered isn't in the dictionary, and dictionary.com just said there's no dictionary results. So the idea, even though this term was coined many years ago, it's not part of the common vernacular. But as I set up a course that I was teaching a year ago that I'll talk about, I also realized that there wasn't a Wikipedia entry for it. And so I took a foray into learning how to do Wikipedia pages and created this site and have been impressed that just in the last 12 months there's been over 6,000 views, with now over 800 a month coming through. And as you are familiar with Wikipedia, this is something that users are able to edit and modify and expand on it. And I encourage many of you on this webinar who have an interest in this to go in and embellish what's already there. I think the more we can keep this updated, while you may think, well, Wikipedia is not a real science source, with 6,000 views, obviously people are looking at it, and we want to make sure that we have the best science represented on this resource that is used worldwide. So I'm not the first person to think about this concept, and actually many before me have done a lot of very critical work in thinking about this. Obviously the group at Berkeley has been a leader in this area in discussing this idea of like the top-down and bottom-up analysis and the sort of factors we want to look at in characterizing the exposome. And I think many of you are familiar with this work by Rappaport and Smith. This figure here by Toby, I really like this. It was a study about looking at metabolomics, but he provided this diagram of the types of exposures we face throughout our lifespan. And as you see there in number 1, you have in utero exposures, which when you think about it in proportion to our body weight, these exposures are very high. They're occurring at a very critical point of development and can have a huge impact on our health. Then as we go along through our life, we may obtain a job at some point and spend many decades in a career that gives us high exposures to some type of chemical. From a clinical standpoint, we may be treated with antibiotics for a condition, maybe chemotherapy, and as we get older, we tend to take more and more medicines and more interventions. Over time, we'll also have the accumulation of environmental exposures of lipophilic compounds, many of the persistent chemicals that, while they have been banned for many years, they still persist in the environment and in our bodies. And from a seasonal standpoint, you can think about things like pollen. Here in Atlanta, these spikes are quite a bit larger. We're over 2,000 on the pollen count today. You have these sort of continued exposures that are rippling throughout time. The challenge is that if someone's sitting here at 45 years of age and you're looking at analysis, you're trying to capture the effects of all of these across time. And this is where it gets in this challenge of this sort of lifespan of exposure, and how do we go back and learn about what happened 30 or 40 years ago. The exposome hasn't been embraced by all. There's been quite a few critical items about it. In this one here, the question was, does the exposome promise too much? And while supportive of the concept, this group suggested that if we focus on biological samples to measure exposure assessment, that there'd be a number of limitations. That the idea of having some sort of magic blood test that you can figure out what everyone was exposed to throughout their lifespan is limited, and I'll come back to why some of those concerns are there. There was a very good meeting sponsored by the National Academy of Sciences looking at exposure science, where some of the earlier thinking on the exposome occurred. One of the ideas expressed in the report was the concept of an eco-exposome, and the idea that there had to be a complementary measure of the exposures occurring outside of our body to interpret the findings from exposome research. And importantly, because you can't reverse engineer solutions if you don't know where the exposures are coming from. But this also presupposes that the exposome is going to be solely a biological in-body measure. And I'm not convinced that that's the way to do this. So, as if the exposome isn't grandiose enough, what I've been thinking a lot about how when you think about all the sort of non-genetic factors in our life, that you start thinking about what the nurture side of the nature versus nurture equation, right? Our diet, behavior, the environment, any other external forces and stress, they leave quantifiable marks on our biological systems. And it kind of made me wonder, does the exposome represent a biological index of nurture, or at least some portion of it? Now, many people may ask, why make it even more complicated trying to address something as huge as nurture? And initially, the thinking is that it would be much more complicated. It's adding in this layer of complexity that isn't necessary. But I would argue that when you start thinking about it as a way of assessing nurture, it goes away from being this sort of esoteric concept in exposure science and puts it right in the middle of biology. So people in the areas of health and biomedical sciences, when you start thinking about nurture, I mean, this is something and a way of addressing how these external factors come into play to developing makes it just much more accessible to all in the field of science. And so while it may make it more complicated, I think it makes it more accessible to all of those in the field of biology and science. So this is where Dean and I had been thinking about this for a few years about, how do you bring in this concept that makes it even grander? And so we recently published an editorial in Toxicological Sciences titled The Nature of Nurture, and the idea was to provide a bit of an update to Dr. Wild's original definition. And just briefly, the cumulative measure of environmental influences and associated biological responses throughout the lifespan, including exposures from the environment, diet, behavior, and endogenous processes. And just to break this down a bit, I'll show you that there was quite a bit of thought that went into this definition, but the first part was that this idea that this is a accumulation of effects. And so we have the word cumulative right in there, and it's referring to both the exposures and the responses. This is a measure. This is a quantifiable entity. We're not talking about hypotheticals. We think the exposome should be something that's truly measurable. The environmental influences are all-encompassing, and we think one of the more important additions to our definition is this concept of associated biological responses. How the body responds to the external insults is as important as the external factors themselves. And things like epigenetic changes or DNA adducts or alterations and stress hormones, the sort of allostatic load concept and defense mechanisms are all part of this associated biological response that can be measured well over time. Again, we think about exposures from pollutions, allergens, infectious agents, both the nutrients and the toxicants we introduce through our diet. Also adding the concept of behavior, which brings in the physical activity, the mental activity, and social determinants of health in a very broad way of defining that. And also importantly, that we are concerned about the exposures that occur from our endogenous processes. So we're a biological reaction vessel. We have metabolism occurring all the time. All our cells are very active. The microbiome is transferring and metabolizing different compounds. And so we also need to be aware of how our bodies are processing these chemicals and what chemicals they may be producing on their self as part of normal metabolism. So we think while this definition, I think it's a more operational definition, that these are things we can actually address. And just to simplify it more, you can kind of cut out the extra phase, is that just looking at the exposome as the cumulative measure of environmental influences and associated biological responses throughout the lifespan. And so again, you're making sure that that associated biological response is a really key part of that. It was interesting, NIEHS recently released an RFI for input on the development of a language standard for environmental health sciences. And I think this is very critical for exposome research. We need to have some common terms and phrases for what we're trying to assess. And I would argue having a more firm definition of the exposome is a key part of that language standard. So just to come back to our gun analogy, I prefer this view, is that genetics loads the gun and the environment may or may not increase the likelihood of the trigger being pulled, but the environment could also influence whether or not the bullet remains in the chamber, or in the event that the trigger is actually pulled, the environment could still influence the path of the projectile and even be involved in the response of the impact. The point here is that the simplistic idea that genetics loads the gun and the environment pulls the trigger is this not a terribly useful analogy, and I think it'd be better if we didn't use it. And just to go on, that the environment is not a loaded gun. It's a compilation of factors that can be manipulated to benefit human health, and I believe it's much more alterable than our genome. This little graph here is something that I've used in speaking to community groups and in the classroom, in that if we think about our various exposures, you can plot them on a graph where you look at things that are somewhat under control at the individual level and things that are controlled at a population or societal level, and you have things that have major effects and minor effects. And so we know as individuals we control things like our diet and smoking, our physical activity, and various behaviors, but things like the quality of our air and water depend much more on a population or government side for regulation. But when we think about things that have major impacts on our health, things like an infectious agent, could be one of the major drivers of our health, and it's a combination of proper behaviors at the individual level, but also things like the development of vaccines by companies and distributions of them. We have things like accidents that have a combination of individual levels and regulations that can prevent that. But the idea of this sort of graph is that we see that there's these things that as individuals we have some level of control over. We see where we might have to go and enlist the help of a government entity to do something. But if you can kind of look at your overall exposures in this sort of graphic, that when something comes on TV that says, hey, blueberries are good for you, well, you know that that's part of this diet that has always been a major driver, and you don't need to start processing this as a new set of systems, is that these sorts of environmental factors are the things that we should be focused on. I put organic food down here as minor and thinking more of it at a worldwide level. For those that have the luxury of being able to purchase organic food, sure, it's great to minimize our exposure to other chemicals, but eating a healthy diet is a much bigger driver to our health. And I would rather have people eating much less fast food and much less red meats and things than focusing on the organic portion of it. I think there's many more things we can do in our diet that are much more effective to improving our health. And so this idea of thinking about how we can change our environmental health protection is something that's been discussed at very high levels. Dr. Collins discussed this idea of transforming environmental health protection. And in talking about some of the concepts of high throughput toxicology, to get more information on what we're doing, and I think these are important factors when we think about the exposome, is we have not done a very good job of measuring the thousands of things we're exposed to. And our traditional ways of doing this have focused on one chemical at a time, and that's just not how we're exposed to things. And the concept of the exposome puts in this sort of the complex exposures we face, which is much more what happens in reality. So I do think that as we drive forward with the exposome, that the main components of environmental health sciences are key. And I think this is something that does need to be driven by environmental health sciences. But it has to include the population level studies done by epidemiologists. It has to bring in the exposure science to know what is out there in the environment. But it also needs to include the biological mechanisms. It has to be focused on the pathways that are involved in controlling our systems. And then all this information needs to be brought into some sort of computational platform where you can integrate these various sub-disciplines to drive forward to the exposome. And so this is something that I've found very interesting in the standpoint of education and environmental health sciences. It's something that does bring together the field rather than fracture it into different groups. And so this was some of the motivation when we started thinking about applying for a P30 Core Center from the National Institute of Environmental Health Sciences in the United States. There are currently 20 centers across the country, and the idea is to provide support for equipment and facilities that are shared among researchers that are looking at various environmental health questions. And these aren't at all focused on the exposome per se, but the idea is you're trying to bring together a group of investigators across a campus, and I thought that that the exposome represented a great concept to bring together the people we had on population studies all the way down to the molecular level. So we put together an application called Health and Exposome Research Center, Understanding Lifetime Exposures, or HERCULES. I'm a big fan of acronyms, and I think when you're trying to stand out on a campus that has many other centers, you need to have a name that does that. And the idea was to promote the importance of the environment in human health and disease using the platform of the exposome. And this is something we're trying to do, not just on our own campus, but throughout. Shortly, about 2 weeks after our grant was funded, I was at the Vatican and came across this bronze of Hercules, and I couldn't help resist incorporating it into some of our work. Our center includes investigators in the School of Public Health, the School of Medicine, School of Nursing, the undergraduate college, and at Georgia Tech. So it's really bringing in a lot of people from, I believe, 20 different departments. So the basis of our center is it's about course to support our research. And so one of the things we thought was very critical in this is addressing the complex data that we have. And so we have a course for systems biology and bioinformatics. The systems biology aspect is directed by investigators at Georgia Tech. There is this integrated health science facility core, which has 3 components. Dana Barr runs our analytical chemistry facility. Dean Jones runs our metabolomics, and Tom Ziegler helps with transferring this into patient and clinical type populations. We're able to give pilot awards each year for people that have innovative ideas on how to address these complex environmental health problems. Paige Tolbert, who's also the deputy director, runs our career development core and trying to encourage people to pursue some of these high-risk aspects and provide additional training if necessary. And then also we have a community engagement and outreach core that Michelle Kegler and Melanie Pearson run. And this has been a really great component to this. I was very concerned about how the community would embrace a concept as odd as the exposome, but they seem to say that they've known this for a long time. They know that it's not just the chemical in the creek down the street or the pollution outside. It's all of these factors. It's their diet, it's their physical activity, it's their medicines. But they've always been challenged with how you keep track of all that information. So the community's really embraced this concept, and we've enjoyed providing some of that information to them. Just to give you an example of some of the high-risk type pilot projects that we have supported, Mark Prousnitz at Georgia Tech recently developed a microneedle patch to deliver flu vaccine, and we started talking to him a few years ago about the idea, could you use these microneedle patches to actually draw interstitial fluid from an individual, and that could provide enough sample to run metabolomics on it that would allow you to look at up to 20,000 different chemicals in a single drop of biological fluid. Dean Jones has really advanced his computational analysis of his metabolomics and is measuring well over 20,000 different chemicals and, again, 50 microliters of blood. And so the idea is that could we move this forward where you might have a situation where you could send out patches to 100,000 people across the country, have them wear it for a day, send it back, run metabolomics, and rather than trying to guess what people have been exposed to, actually measure what is in their body. So again, trying to advance some of the technologies that we think we would need to do to really perform some sort of population level studies to get an idea of how various exposures affect health. We also have tried to really promote the concept. We have a website for the HERCULES Center that we have been developing and hope to kind of keep this very up to date and a very informative place for people to learn about what we're doing at our center. As I mentioned, our community engagement core has been very successful. Our scientists have gone out into the community. Michelle Marcus, shown here in this picture, is an epidemiologist who has been looking at a group of polybrominated biphenyl exposed people in St. Louis and using some of the approaches that we have in our center to expand her epidemiology studies to bring in some of these exposome concepts to see if we can address how these various exposures are affecting the individuals in the community. We also developed a website called the Human Exposome Project. This is just trying to gather interest and updates on what's happening in the field. We hope to continue to expand this. It's very much I hope it to be a shared project while we're running it here out of Emory. It's trying to have more of a general open effect. I think that's something we can really build out to complement things like the Wikipedia site to provide important news about what's happening in the field, and I hope to kind of use this as an outlet for all the things other people are doing across the world. Other things that we've done, I was never that much into social media, but my webmaster said a lot of people are using this. You need to get up to speed. So we have Facebook and Twitter accounts for both our center and the Human Exposome Project. And this has been growing quite a bit. The number of followers you see there is when we started, it's gone up much more, and I think it's really helped with some of the circulation, and I think it's something that we need to try to reach out to any groups we can, and many of the younger among us and the teenagers and college students are very much embracing these technologies, and I think it's important for us to get our message out to these areas. One of the outgrowths of our center was last year we developed a course called Genome, Exposome, and Health. We didn't think a class on the exposome would work because no one really knows what the exposome is, but sneaking in exposome between genome and health seemed to work. We've had people sign up for the class. Not just our Masters of Public Health students, we've had many upperclassmen in health-related majors and doctoral students in environmental health sciences. And so the idea here is to talk about the sort of state-of-the-art of where we are with genomics and epigenetics and remote sensing, exposure assessment, bioinformatics, systems biology, and how we start pulling some of these things together. Here's an example of some of the topics we have. So we, each week they hear from an expert in the field about these topics, and then we get into the very recent literature and have them discuss how these technologies are being used to advance our understanding of health. And so this has been a very enjoyable class. It's been a little bit intimidating because we're trying to teach something that we don't totally know how to do yet. But the students seem to be interested in our attempts to do so and have been very patient with us as we develop it. And so again, we're very interested in the enabling technologies and how we integrate these things, and the data management is something that we've also talked quite a bit about in the class. Some of the examples and analogies that I've used in the class I think have worked well for people in the community. This is something that I've referred to before, is that if you imagine that you'd had some really good chicken soup at some time and wanted to recreate that soup, if you just had a sample of the soup, you'd be very hard-pressed to do that. If someone just gave you a list of the ingredients, you'd have a very hard time recreating that perfect soup if you don't know how they prepare the ingredients and what order they put them in. If you really want to reproduce what's there, you need both. You need to know what's on the outside, and you need to know what's in the soup itself. Well, our biological soup isn't very much different. We're exposed to all sorts of different chemicals and nutrients and things that come into this biological soup, and it cooks in our bodies for years. And if we want to figure out how these things, these chemicals on the outside, are affecting our biology inside, we need to measure what's going on inside the soup, but we also have to measure what's happening outside of our bodies, because if we think a certain chemical is involved in a disease, we have to be able, again, to reverse engineer the solution, which goes back to the source of these chemicals to reduce that. After I started teaching the class last year, about a year ago, I was at the Society of Toxicology meeting, and the students had kind of been concerned there wasn't really much background material. And so I talked to people at Elsevier and Academic Press about writing a short book on the concept. They liked the idea, and in June we signed a contract, and 3 months later I sent them the final draft of this book. And this was something that was a very intensive but short effort to kind of put together some information from my view of this. I hope that it's not the last text on the exposome. This was just something just to kind of get the idea out and get some background information. But I think that we do need to think as a field about a more comprehensive text once we kind of have a better idea of where we're going with this, because it is a complicated topic and it needs some very detailed information available to those that want to learn more about it. And so in thinking about where we kind of go with the exposome, one of the things we've talked about is that, do we need to avoid what others are already measuring? I talked about looking at epigenetic changes due to an exposure. The question is, should we be focusing on just the chemicals themselves because other people are looking at epigenetics and stress responses? But at the same time, that some of the enduring biological responses to some chemicals may be the only evidence we have of a transient exposure. If you're exposed to some short-acting chemical a year ago, the chemical itself will be long gone, and all that may be left is whatever that response was. And so this is something we're going to have to think about quite a bit in the field to kind of how we kind of make our mark. One thing that I've considered, and this is very much a rough idea, but the idea of having some sort of exposome index that addresses what we think is unique to the exposome. So the idea, if we could measure the deposition of persistent organic chemicals, a nutrient profile, P40 variants, P450 variants, the actual exposures that occur, looking at some of the key biological responses, for example, DNA adducts or methylation, telomere integrity, or other indices of allostasis. The behavioral factors, things like physical activity, location. Many of these could be tracked by GPS in your phone. And then other key exposures such as exposure to infectious agents, residual antibodies from such exposures, pollutants, and pharmaceuticals. But the idea of if we could come up with something that could be measured in a uniform way, I think that would be an advance. Again, I don't know what that list would look like. This is just something kind of for discussion's sake. As we think about the sort of complex data we would be collecting, and when we go from thinking about the sort of biochemical pathways, how the physiological pathways are affected, the data we would get from human studies and omic studies, population data, and the environmental data from exposure assessment, we have data coming from across all these different areas, and we have to decide which are the ones that are important, and then how do we start merging and integrating these things together. Again, I don't purport to have the solutions. I just see these as being areas of need. So again, I think the idea of data integration is critical. We have a lot of sophisticated technologies to measure the chemicals, and many more are under development. But I view that we're lacking the bioinformatic or systems biology approaches that we need to assimilate these types of data into comprehensive models. As a biologist, I'd very much like to see something that has the sort of physiological pathway-based model for the exposome. It's why we think certain things, if we think it's affecting a certain pathway, if we think it's affecting steroid receptors, cell death pathways, various nutrient pathways, Krebs cycle type things, that bringing these things back to the biology is an important thing to do to make sure that we're not just looking at associations, but actually doing things that can be analyzed and interrogated at a computational level later on. I put this in my book, this sort of rough timeline of what I thought would be done for a human exposome project. And the idea that right now I view us as being in this sort of 5-year preparation stage. We have many of the ongoing projects that are occurring now that we're going to learn from some of these pilot studies. We're going to learn from these larger studies that are occurring in Europe, which I'll mention later. But after a few years, we need to kind of go through and evaluate what the best measures were, the best outcomes to measure, and then really begin a more coordinated and rigorous study where multiple sites are looking at multiple populations in a very consistent format. And that project, it may take 5 years, it may take 10 years, but it's something that needs to be done in a very methodical way. It's something that we're not going to get started next week. But at the same time, our European colleagues are way ahead of us. The European Union and others have made some major awards to look at the exposome. Hopefully many of these people will be speaking in future webinars, but the Exposomics project includes many people in the UK and Europe. The HELIX project is looking at some of the early life exposome aspects, and they've really jumped right into this and are doing major studies right now. The HELIX project, again, is talks about this concept of nature versus nurture, which is nice, because I like this sort of the nurture aspect of it. But so again, this is, I think, it's going to be really great because the European Union has invested in this, and these groups have gone into this, which to me feels a bit intimidating because I wouldn't even know where to start with this. But obviously, these groups have come up with some great ideas, and I think it'll be very exciting to see what they do, and it's important for us on the U.S. and NIH side to look at their findings and to see what we can learn from them. So, in just kind of an example here of how the exposome can help with some of these concepts is we have all sorts of studies from GWAS, from these genome-wide association studies, but we really need to think about how we can go past that. And just to give an example, again, I do research on Parkinson's disease, and a recent GWAS was conducted that identified 2 well-known genes in Parkinson's disease and also identified a new one involved in antigen presentation. And the group published that paper and then continued to work on things. And one study that was very exciting was that they started looking at some of the, in a way, very simplistic environmental factors. So they had data on caffeine consumption, they had data on ibuprofen use, they had data on smoking status. Very strange effect is that smoking is protective in PD. There's dozens of epi studies that demonstrate this. No one knows why this is. Well, this study that added in this layer of smoking or nicotine into this GWAS pulled out some SNPs that appeared to mediate this effect. And so if you had the major alleles, cigarette smoking was protective, as we've always seen. But if you had the minor alleles, smoking actually made your Parkinson's worse. And I was very intrigued by this, and they identified a gene called SV2C. And for those of you that know much about the brain, if you see where this SV2C is located, it's right in the pathways that are affected in Parkinson's disease. And so I was very intrigued by this. And we were also interested in that as a toxicologist, we knew that botulinum neurotoxin A actually gets into neurons by hijacking along with SV2C. And we knew that its family member SV2A was a target of a blockbuster drug for epilepsy called Keppra. So the idea that this might be a druggable target was very interesting to us. Just real quickly, we started making several tools that we could study SV2C, antibodies, doing localization, making knockout animals, working with optogenetics and trying to target the release of certain things. And just real briefly, here we are just over a year later. We have recently identified that SV2C is a novel regulator of dopamine release. And so this is a discovery that would not have occurred by the GWAS alone. Until they brought in the one environmental factor of smoking, we wouldn't know what we know now on this. And that's just with one environmental factor. So I think that illustrates that we may not need to measure everything. If we can just provide a valid set of parameters for the environment, for that E, that can be used in conjunction with existing studies that employed genomics, epigenomics, and proteomics, et cetera, that we could really advance some of these studies. And I think it's also important to remember that one of the goals here is to provide a means for the non-environmental health scientist to incorporate the E into their studies. Even if it's only occurring at a single time point in an individual who's 45 years old, if we can get that level of measure that, again, that non-environmental health scientists can add into their studies, I think that should be a prime deliverable for any sort of human exposome project that something like NIH would fund. And it may be that a few international cores could provide this service. Maybe that people that are doing large GWAS studies, they send samples to this group where they can do a systematic analysis of various environmental chemicals and measure that exposome index, and do it at that level of the Human Genome Project where you can process thousands and thousands of samples, and that the need and demand to do it drives the technology forward to allow us to do things that we just didn't think were possible more recently. And a great example, I'm sure Chirag Patel will participate in one of these webinars later on. He published the first environment-wide association study, and he used the NIEHS data to do this. And I think while the NIEHS data provides a lot of very useful information, I think we can do better than that. I think we can provide better information on environmental exposures that would provide even greater power to these sorts of approaches that Chirag and some of his colleagues have used. So I want to end here with an analogy that came across me very recently. So the Society of Toxicology meeting was in Arizona last week. A week ago today, my wife and I hiked to the bottom of the Grand Canyon and back in a single day. And I was struck by the similarities between the start of our trip and where we are with the exposome at this point. We are at the beginning of a rocky and unsteady journey, and it's not going to follow a linear path. You see up here in the corner, these are the switchbacks you use to get up and down out of the canyon. People will tell you not to do it. They'll tell you it's too hard. They'll tell you it's too dangerous, and they'll tell you that you're crazy. And there's some things we learned from this. It took longer than we thought it would. It was much harder than we thought it would be, and it hurt more than we thought it would. And if you look here at this bighorn sheep up on the hill, that's him right there, you see a slight misstep could be tragic during this process. And what's important to remember, though, is that it was more rewarding than we anticipated. So even though the trials and tribulations, it was, again, much harder, it was much more rewarding. But important to remember that us going down to the river at the bottom was an option. We made a decision to do that. But getting out of the canyon and completing the journey was not an option. We had to get out. And I think that's something we need to think about as we move forward with the exposome, is that we need to be very methodical as we start the journey. But once we start it, for the sake of the environment, environmental health sciences, it has to be done well and it has to be done correctly. And while the views were spectacular, we did encounter mule urine and feces along the way. And I think this is an important thing for us to remember as we pursue this, because it's not going to be easy. There's going to be a lot of troubles. But I do think that it's time to start planning this journey. And as we can discuss some more, the Exposome meeting is planned for January next year the National Institute of Environmental Health Sciences, and I hope this becomes the start of a major initiative that not just includes NIH in the United States, but uses our worldwide partners to really make some major advances on the exposome. And with that, I will turn it back over to our moderators.